Hello, welcome to Notable Nobels, a podcast about the Nobel Prize in Physiology or Medicine. My name is Harrison Doolin. I'm a postdoctoral scientist at the Fred Hutch Cancer Center in Seattle, and I will be your host for this web series. The purpose of this series is to trace key advancements made in the biological and medical sciences over the past century and a quarter, and we're using the Nobel Prizes in Physiology or Medicine as a guide. Now, every career has its highest prize, and for scientists, that prize is the Nobel Prize. It's the most prestigious award a scientist can receive, and it marks discoveries that have made a profound impact on our understanding of biology and our ability to treat diseases. Now, just to give you a little bit more of an introduction this time, we are getting towards the end of what I'm going to call Season 1 of this podcast, which has focused on infectious diseases. We have spent a lot of time recently focused on viruses. I think the last 10 episodes have been virus-related. Now, we aren't quite done with viruses yet, but we're going to shift gears for the next 3 episodes. We will now be turning our attention to antimicrobial drugs. My goal in the next 3 episodes is to build on previous episodes, particularly the antibiotic episodes, so if you haven't listened to those yet, go have a listen. And I hope to provide you with a feel for how the process of drug discovery has changed over time. For today's episode, we will be examining one half of the 2015 prize in Physiology or Medicine, which was awarded to the rather extraordinary Tu Youyou. Tu has the distinction of being the only Chinese individual to date to win the Nobel Prize in Physiology or Medicine, and she's also the only Chinese woman to win a Nobel Prize in any category. The Nobel Assembly at the Karolinska Institute chose to give Tu the award, quote, for her discoveries concerning a novel therapy against malaria, unquote. Today we will be going over Tu's work in herbal medicine, how she translated that work into a powerful anti-malarial drug, and I'll be ending the episode with a word about the natural medicine movement in the United States. But first, let's look at the remarkable background of Tu Youyou. Tu Youyou was born in December of 1930 in Ningbo, China. Her father was a banker, her mother was a stay-at-home mom, and Tu was the only daughter among the family's 5 children. It was a tumultuous time to be growing up in China. The country was plagued by Japanese and Western imperial forces from without and civil war from within. Nevertheless, Tu was able to do well at school and made it through her middle school studies without incident. However, when she was 16 years old, she contracted tuberculosis, which forced her to take a 2-year break from high school. The incident inspired her to study medical research after high school, so in 1951, she entered the pharmacy program at the Medical School of Peking University, now known as the Peking University Health Science Center. It was during her time at university that she was first exposed to the discipline of pharmacognosy, which would later become the basis for her Nobel Prize-winning discovery, but more on that in a minute. Tu graduated from university in 1955 and was assigned by the government to the newly established Academy of Traditional Chinese Medicine, now known as the China Academy of Chinese Medical Sciences. Tu would spend the rest of her career at the academy, and it was there that she would make her Nobel Prize-winning discovery. So, what was Tu's role at the academy? Well, she was working in the fields of pharmacognosy and phytochemistry. These disciplines are not very well known, so let's define them. Pharmacognosy is the study of medicine from natural sources such as animal parts, herbal plants, or medicinal fungi. The drugs derived from these sources are often called crude drugs because their biochemistry and mechanism of action are usually not well understood. However, the goal of the pharmacognosist is to take these crude drugs and study them scientifically with the goal of developing the crude drug into what we might call a modern drug, with a fuller understanding of the molecular biology and biochemistry behind what the drug is and how it works. Phytochemistry is the study of chemicals made and derived from plants and includes the isolation, purification, and molecular identification of plant chemicals, as well as studying the interaction of these chemicals with other species. A lot of the work of phytochemists is what we would call fundamental research, or research done simply to understand something for its own sake. However, in China, in the middle of the 20th century, there were very few resources available to spend on fundamental biology questions. Scientific projects were restricted to applied research, or research done to find a practical solution to a problem. Tu's work was therefore restricted to the identification of drugs from traditional crude plant medicines that might be useful for fighting diseases. During her first decade of professional work, Tu researched the effectiveness of herbal medicines against parasitic worm infections, and she also took a 3-year break from research in the late 1950s to participate in a training program in traditional Chinese medicine. Then in the 1960s, scientific research began to slow. The Chinese Cultural Revolution was in place, which had a devastating effect on scientific research in China. Lots of researchers critical of the government were sidelined, and many chose to leave the country. However, the government still needed scientists to come up with solutions to the practical problems it was facing. One of those problems was malaria and its impact on the Vietnam War. The Vietnam War, which began in the mid-1950s, slowly became an international conflict and a key part of the Cold War between Eastern Communist nations versus the Western Bloc. Initially, the two sides supported their proxies in Vietnam mostly financially, but in the 1960s, the on-the-ground military presence had dramatically increased. Between 1959 and 1964, the US military presence in Vietnam grew from less than 1,000 troops to over 23,000. Then in 1964, US President Johnson began a rapid escalation of troop deployments to Vietnam. By 1968, over 500,000 US troops were in Vietnam. In response, China began sending tens of thousands of their own troops into northern Vietnam, starting in 1965. And by 1967, over 170,000 Chinese troops were in Vietnam. As the war dragged on, both sides were looking for every possible advantage over the other. A persistent problem for both sides was troops taken out of commission not by bullets, but by malaria. Malaria had been a known problem for decades in both the West and the East, and there had actually been a lot of success in combating the disease in the first half of the 20th century. But cases of malaria began ticking back up again in the 1960s. To understand why that was the case, let's take a few minutes to talk about malaria, what it is, how it makes you sick, and some of the history of trying to prevent it. Malaria is a mosquito-borne disease caused by a single-celled parasite. The parasite enters the bloodstream of the infected host after a mosquito bite, where the parasite is transported to the liver. In the liver, the parasite infects liver hepatocytes and begins to replicate. New parasites are released into the bloodstream, where they infect red blood cells. The parasite will replicate inside the red blood cell until the cell bursts, releasing the new parasites into the bloodstream. These newly released parasites can then go on to infect more red blood cells. These parasites in the blood can be picked up by another mosquito when it takes a blood meal from an infected person, which starts the malaria life cycle all over again. Most people infected with malaria will get symptoms of fevers, chills, and headaches before their immune system clears the pathogen. But a subset of patients develop severe malaria, which has multiple causes stemming from infection with the parasite. One of the most obvious problems is the death of red blood cells. Red blood cells carry oxygen to all of your cells in your body, and they remove CO2. The loss of red blood cells during malaria can make a person anemic, and as cells throughout the body lose access to oxygen, those cells will start to die, which can lead to organ failure and death. Additionally, the bursting open of red blood cells releases toxins into the blood, which are normally cleared by the kidneys. However, a massive amount of red blood cell death all at once, like what happens in malaria, can be toxic to the kidneys, and malaria patients can develop kidney failure. If that weren't bad enough, malaria-infected red blood cells lose a lot of their flexibility and become stickier than regular red blood cells. This results in the sequestering of red blood cells onto the lining of blood vessels, which can cut off blood flow to the organs. When this happens in the brain, this can result in a condition called cerebral malaria, which is a life-threatening form of malaria. Now, as discussed on previous episodes of this podcast, once people discovered mosquitoes were how malaria was spread from person to person, anti-mosquito measures were put in place to help stop the spread of malaria. This included things like insect repellents, insecticides like DDT, and mosquito netting. Additionally, there was effective drugs to help combat malaria, and the most widespread drug used against malaria was chloroquine. Chloroquine was a chemical derivative of another malaria drug called quinine that was widely used in the 19th century. Chloroquine came into use after World War II and was shown to be an extremely effective drug. It became standard practice for Europeans visiting malaria endemic areas to take weekly chloroquine pills to protect against malaria. With all these successful measures in place, scientists thought that the threat of malaria was over. However, starting in the 1960s, malaria cases began increasing again. In the war in Vietnam, despite regular prophylactic treatment of soldiers with chloroquine, around 40,000 American troops developed malaria. Some of these infected troops brought the parasite home with them when they returned to America, which in the late 1960s led to pockets of locally spread malaria cases in the US. This was rather alarming to epidemiologists, considering the US had declared endemic malaria eradicated back in the 1950s. So what was going on? Why was malaria increasing? Despite the anti-mosquito measures and the use of chloroquine, why were tens of thousands of soldiers still getting sick? The main answer was drug resistance. The widespread use of chloroquine helped select for the development of chloroquine-resistant malaria strains. As a result, malaria cases began to creep back up, and the soldiers in Vietnam, on both sides of the conflict, were left without an effective anti-malarial drug. Both China and the United States appreciated that developing a new effective anti-malarial drug would give them a leg up in the Vietnam War. So, in secret, the militaries of both governments began recruiting scientists for research programs to develop new anti-malarial drugs. The U.S. research program was based out of the Division of Experimental Therapeutics at the Walter Reed Army Institute of Research in Washington, D.C. The Chinese research program, named Project 523, recruited scientists from several institutions in China, including the Academy of Traditional Chinese Medicine in Beijing, where Tu Youyou was working. And so, in 1969, Tu Youyou began working for the Chinese military to try and find a new anti-malarial drug. So how did she do that? How did Tu go about developing a new anti-malarial drug? Well, Tu was a pharmacologist working at the Academy of Traditional Chinese Medicine, so she decided to screen traditional Chinese remedies to see if any might be effective against malaria. In the first few months after joining the government's anti-malaria project, Tu and her team combed through traditional Chinese medical texts and folk recipes and spent time interviewing experienced Chinese folk doctors. The result of this work was over 2,000 herbal, animal, and mineral crude drug recipes, which Tu summarized and distilled into 640 different anti-malarial prescriptions. Now, 640, that's a lot of potential drugs to go through, but Tu and her team decided to give it a try. In 2 years, between 1969 and 1971, they tested about 100 of these different prescriptions in a rodent model of malaria to see if any of the drugs could be effective against the parasite. However, none of them worked. After these 2 years, Tu decided to go back to the traditional Chinese medical texts to see if she might have missed something. One of the ancient books she turned to was titled A Handbook of Prescriptions for Emergencies by the Chinese court official and physician Gi Hong. Gi Hong had written the book back in the 4th century, and in it, he described some traditional Chinese remedies for relieving malaria-like diseases. One of these remedies used the herbs from the Artemisia family, which in Chinese are called Qinghao. Gi Hong's Qinghao recipe said to do the following. Take a handful of Qinghao, immerse in 2 liters of water, wring out the juice, and drink it all. Now, Tu Youyou lingered over this recipe. Her lab had tried using some of the different prescriptions that used the Qinghao herb, but they'd gotten mixed results. In the cases where the drug seemed to have worked, they'd been unable to replicate their results. Lingering over Gi Hong's recipe, Tu noticed that Gi Hong said the herb should be wrung out in water. This was different from a lot of the recipes they'd tried so far, which said herbs should be boiled in water. Tu hypothesized that their failed attempts at using Qinghao had been because by boiling the herb, they'd accidentally inactivated its key ingredient. Going back to her lab, Tu decided to try making extracts of the Qinghao herb using lower temperatures and a few different solvents. She used water like the original prescription recommended, but she also tried a few organic solvents, including ethyl ether. When Tu's lab tested the different Qinghao extracts in their rodent model of malaria, they found that the ethyl ether extract could inhibit malaria 100% of the time. Tu and her lab modified their extraction technique a bit to get a purer form of the drug. Then in 1972, they tested it in malaria-infected monkeys. The drug was again shown to be remarkably effective against malaria. Tu presented her lab's findings at a scientific conference in Nanjing in 1972, and the results generated a lot of interest among the Chinese scientists. Moving quickly, the first clinical trials with Qinghao extract took place in Henan Province, China, between August and October of 1972. In a small study, 21 patients, the drug was deemed safe, and encouragingly, patients that received the new drug had rapid recoveries while patients that received chloroquine did not. Things were progressing pretty nicely. But it was at this point that Tu would show herself to be not merely an herbalist with a great plant, but a truly modern scientist. Tu and the other Chinese scientists used the latest chemistry techniques to obtain a highly purified powdered form of the active ingredient of Qinghao. They named this new drug artemisinin, using the Western name for Qinghao. Using her skills as an organic chemist, Tu began synthesizing derivatives of the naturally occurring artemisinin drug to see if any of the new chemicals might show even better efficacy against malaria. One of her derivatives was dihydroartemisinin, which she synthesized in 1973. Dihydroartemisinin was found to be 10 times more potent than artemisinin against malaria. For the rest of the 1970s, Tu and her team, as well as other Chinese scientists and clinicians, continued to work on artemisinin and its derivatives, both in the laboratory and in the clinic, and it proved to be a very successful drug. Now, all that sounds really fantastic, right? But since this is an episode about the 2015 Nobel Prize in Physiology or Medicine, you might be asking, well, why did it take so long for Tu to be recognized for her achievement? If the drug entered use in the 1970s, why the long delay? Well, there are several answers to that question, and all of them are fascinating. First of all, Tu's work was technically a secret government project, so she was only sharing the results of her work with people associated with the project. She wasn't allowed to share it with anyone else. Secondly, all of this work was taking place in the middle of China's Cultural Revolution, which was aimed at strengthening communism in China. Communism, by definition, recognizes the commune or the community, whereas prizes like the Nobel Prize are typically awarded to individual scientists. During the peak of the Cultural Revolution, Chinese scientists were actually forbidden from publishing their work since it was seen as too individualistic. While Tu was able to present her work to her fellow government scientists, a lot of the initial work on artemisinin went unpublished until after Mao's death and the end of the Cultural Revolution. And thirdly, let's remember that this period of Chinese history was also taking place in the middle of the Cold War, and the impetus to develop artemisinin as a drug was the Vietnam War. For almost all of the 1970s, the US didn't officially recognize the People's Republic of China as a nation, and the West in general was extremely anti-communist. It wasn't until 1979 that the US formally recognized the People's Republic of China as a country. This meant that for years, nobody in the West had any idea that a brand new, highly effective treatment for malaria had been developed. As tensions between China and the West diminished at the end of the 70s and the beginning of the 1980s, there finally came an opportunity for scientists on the two sides to come together. In 1981, the city of Beijing was selected to host an international scientific conference on anti-malarial drugs. This was sponsored by the United Nations and the World Health Organization. By that point, artemisinin and its derivatives had been used to treat several thousand patients in China. At the meeting, Tu was able to present the data on artemisinin to the West for the first time, and it immediately generated worldwide attention. In 1982, 10 years after Tu's initial discovery, the data on artemisinin was finally published in the Journal of Traditional Chinese Medicine. Artemisinin-based drugs over the next few decades began being used around the globe as a first-line treatment for malaria. The Nobel Assembly took notice, and in 2015, Tu Youyou became a recipient of the Nobel Prize in Physiology or Medicine. So, what is the current usage of artemisinin-based drugs since their discovery and worldwide acceptance? Well, this part of the story involves a very cool bit of poetic irony. We'll start out by saying that the drugs have been a huge success. Over 1 billion doses of artemisinin-based drugs have been manufactured in the last 2 decades, which have been credited with saving millions and millions of lives. Artemisinin-based drugs are used in over 90 countries around the globe, and they are on the World Health Organization's list of essential medicines. However, artemisinin has a catch. It has an extremely short half-life in the body, only a few hours. Fortunately, the drug is cheap to produce and can be manufactured at high quantities, but the short half-life of the drug means it's easy to select for artemisinin-resistant strains of malaria if patients don't stay on their drugs. To compensate for this, artemisinin-based drugs are always given in combination with another anti-malarial drug, usually one with a different mechanism of action and a longer half-life. This form of therapy is called artemisinin-based combination therapy, or ACT for short. ACT is the currently recommended first-line treatment for malaria by both the World Health Organization and the United States Center for Disease Control. There's about half a dozen other anti-malarial drugs that the WHO recommends for pairing with the artemisinin-based drugs to form ACT. Now, one of those other recommended anti-malaria drugs is mefloquine, and here's the cool bit of irony that I'm so excited to tell you about. Remember earlier I said that in response to the Vietnam War, both the United States military and the Chinese military started programs to develop anti-malaria drugs? Well, artemisinin was the result of the Chinese program, and mefloquine was the result of the American program. Now, mefloquine is not as good a drug as artemisinin. It's more expensive and it has more side effects. I'm not at all surprised the Nobel Prize went to artemisinin. Nevertheless, mefloquine is still an effective drug. But like other malaria drugs, if you give it by itself, drug resistance can develop. So, the recommendation by the WHO is that you give them together. Isn't that crazy? These two drugs, which were developed by these two countries to better fight each other, must work together if either drug is going to continue to work at all. And I just think that's a beautiful bit of irony. I love it. I was very excited to share it with you. Okay, we're getting towards the end of this episode. I just have one short tangent to say here as we wrap things up, and it's about the natural medicine movement in the United States. The discovery of artemisinin and Tu Youyou's winning of the Nobel Prize has lent some support to herbalism and other forms of natural medicine, particularly in the United States. The natural medicine movement is rather hard to define since it encompasses a wide range of therapies for a bunch of different conditions. It's also sometimes referred to as alternative medicine to distinguish it from mainstream Western medicine. A common theme found among some, but not all, supporters of natural medicine is a suspicion of modern medicine, particularly medicine marketed by pharmaceutical companies. Natural medicine instead favors, quote, natural cures through things like herbalism and dietary supplements. In its worst forms, the natural medicine movement results in resistance to vaccines, chemotherapy, surgeries, and produces books with titles like How to Stay Healthy Without Pills. That's a book that, full disclosure, I did not read, but the title does exist. In case you can't tell, I'm not a big fan of this kind of thinking. The idea that something is better or worse because it's natural or unnatural needs to be done away with. It's not a helpful paradigm. And I want to hold up Tu Youyou as a great example for us to follow in this. Tu's work undoubtedly shows the benefits of medicines from natural sources. There's nothing wrong with looking to nature for cures for human ailments. But cures from natural sources need to be subjected to rigorous testing. Ideally, the same kind of testing we use for cures from unnatural sources. To show why testing is important, particularly with a lot of these traditional folk remedies, remember that Tu's group tested over 100 different traditional Chinese medicines that didn't work against malaria before they found the Qinghao herb. Nevertheless, Tu still appreciated that natural remedies existed, but in addition to her appreciation of drugs from natural sources, Tu recognized that modern science could improve what nature had supplied with the artemisinin drug. And how did she do this? Well, she took the naturally occurring artemisinin compound and she chemically modified it to produce the unnatural compound dihydroartemisinin, which we mentioned earlier is 10 times more potent than artemisinin. Here's how Tu summarized her work in her autobiographical page on the Nobel Prize website. Quote, "Exploring the treasury of traditional Chinese medicine has provided us with a unique path leading to success, while utilizing modern scientific techniques and approaches are no doubt an effective and efficient way of realizing and expediting discoveries." Unquote. So let's all be like Tu. We can appreciate what nature has to offer, we can look to it for cures, and we should also appreciate what science has to offer, and we can look to science for cures as well. Okay, that concludes this episode of Notable Nobels. This episode was recorded on May 20th, 2024. I want to thank Digital Mind Productions for providing the music and Nicholas Wynn for the graphic design. Next time we'll again be looking at drug discovery, and we'll finally get to talk about pathogens that thus far have been mostly ignored in our survey of infectious diseases. Want to know what that is? Well, here's a hint. Shai Hulud. Want to know more? Well, listen next time to find out. Thanks so much for listening. See you then.